Okay, so welcome to this next video in which we are discussing synchronous and asynchronous neurotransmitter release. Okay, so we're in the process of describing synchronous neurotransmitter release. So far, we've got to the stage where the action potential has arrived and caused the opening of these N and PQ type voltage-gated calcium channels, which is then going to allow calcium into the cell, which is going to go directly onto the machinery of the synaptic vesicles. Okay, and the protein it's going to activate is Synaptotagmin 1. So, let me draw another picture of this docked synaptic vesicle here. So here's our synaptic vesicle, here's our plasma membrane, here's our trans-core snare complex, with our Synap25 here, and our Syntaxin 1 here. So let me colour these in. So in blue here we have Syntaxin 1, okay. In turquoise here we have Synap25, and I realise actually that I should be drawing both of these at the same time so that I can highlight them in at the same time. So here's my other core snare complex. Okay, right. So in turquoise we have the Synap25 T-snare here. Okay. In blue we have Syntaxin 1 protein, which is another example of a T-snare. Okay, and then finally, the 1V snare, which is in orange here, which is Synaptobrevin 2. Okay, so here we have our docked synaptic vesicle at the membrane of the axon terminal. Now, in the membrane of the vesicle, you also have a protein known as Synaptotagmin. Okay, and it's usually Synaptotagmin 1. Okay, so I'll draw Synaptotagmin like this. So Synaptotagmin has these two special domains, which are C2 domains. So this is the C2a domain, and this is the C2b domain here. So this is Synaptotagmin 1. Okay, right. So, I will outline Synaptotagmin 1 in this purple colour here. Now, these C2 domains, they are just protein domains which are capable of binding calcium ions, basically. So, overall, 3 calcium ions are capable of binding to the C2a domain here, and 2 calcium ions are capable of binding to the C2b domain. And when the calcium binds to these C2a and C2b domains of this Synaptotagmin 1 protein, it doesn't cause a conformational change in the Synaptotagmin C2a and C2b domains. Instead, all it changes is the electrical potential of the C2a and the C2b domain. So the electrical potential of the C2a domain goes up when you add these 3 divalent cations on. So you've effectively added 6 positive charges onto here. That causes a rise in electrical potential at the C2a domain. Similarly, when you add these 4 positive charges onto the C2b domain in the form of these 2 divalent cations, that causes a rise in the electrical potential of the C2b domain. Now, when that rise in electrical potential happens, it means that the C2a and the C2b domain can start binding to things. Now, one of the things that Synaptotagmin 1 has been shown to bind to is the snare protein Syntaxin 1. So when the calcium arrives, Synaptotagmin 1 has been shown to bind to this snare protein in blue here, which is a T-snare and is Syntaxin 1. Okay, and you can imagine how that's going to help bring the vesicle maybe closer to the plasma membrane. More importantly, however, the C2a and C2b domains both bind to lipid components of the phospholipid bilayer that are in the plasma membrane and not in their synaptic vesicle. So now let's discuss some of these lipids in the plasma membrane which the C2a and the C2b domain bind to. So we'll go over the page for this. Right, okay, so we'll begin with that C2a domain. So if I just remind you, I'll draw a picture of Synaptotagmin here so that we know what we're looking at here. Okay, so here is Synaptotagmin, and remember, it was in the membrane of the synaptic vesicle like so, and here's the C2a domain closer to the portion which spans the membrane, which is an alpha helix, incidentally, that spans the membrane here. And then there's the C2b domain. And remember, the C2a domain binds 3 calcium ions, whilst the C2b domain binds 2 calcium ions. Okay, now, the C2a domain is going to bind to a component of the phospholipid bilayer of the plasma membrane. Okay, and this component is a molecule known as phosphatidylserine. Okay, right, so in order for me to explain to you what phosphatidylserine is, we first need to know what a phosphatidate molecule is. So I'm going to explain to you what a phosphatidate molecule is. And phosphatidate is the most common constituent of phospholipid bilayers. It's basically the old name for a phospholipid. So, let me show you the structure of a phospholipid. A phospholipid can be drawn as in cartoon form, like so. Okay, so let me just put this sitting in a phospholipid bilayer. So here's the inner and here's the outer leaflet of the phospholipid bilayer. So this is the outer leaflet, which is the layer of phospholipids facing the extracellular fluid, and here's the inner leaflet of the phospholipid bilayer, which is the layer of phospholipids facing the cytoplasm. Now, if we look at this phospholipid in a bit more detail then, it has these 2 hydrophobic tails, as they're called, which I have now coloured in orange. So these here, these things, these are hydrophobic tails, okay? And they are made by taking long-chain carboxylic acids, or also called fatty acids by biochemists. You take long-chain carboxylic acids, which are carboxylic acids which have a very, very long hydrocarbon tail, and you esterify them to the first and second hydroxyl groups of the backbone of the phospholipid, which is a molecule known as glycerol. Okay, so you take this glycerol molecule, which has 3 carbons, each with a hydroxyl group, and to the first 2 carbons, you esterify long-chain carboxylic acids onto those. Now, the proper name for glycerol, the chemist's name for glycerol, is propane 1,2,3-triol. And although that's a bit more of a mouthful than glycerol, which is why the biochemists still use glycerol, this tells you exactly what glycerol is, whereas glycerol doesn't give you any help at all. So it's just a 3-carbon molecule with an alcohol group stuck on each of the 3 carbons. Okay, now to 2 of those hydroxyl groups, we have esterified long-chain carboxylic acids. To the 3rd, what we do is we add this final group here, which is a phosphate group. Okay, so we form a phosphoester bond, or a phosphate ester bond, more properly, with this 3rd hydroxyl group of the glycerol molecule. So this is a phosphate group here. Now, this overall structure that we have drawn is a phospholipid. So this is a phospholipid, equals phospholipid. However, phospholipid has an old name which you probably haven't, isn't used very commonly anymore, but is used in names still like this. And the old name for a phospholipid is a phosphatidate molecule. And if someone was just talking about a normal phospholipid, they would never ever use the name phosphatidate. But when you're talking about modified phospholipids, for some reason it comes back. And this is the origin of this phosphatidyl here. It basically means that this molecule, phosphatidylserine, which by the way is often just abbreviated to PS, it's basically just a modified phospholipid. It's a modified phosphatidate molecule. So let me show you how you're going to modify it. What you're going to do is you're going to take the amino acid serine and you're going to bind it to this phosphate group that's on the phosphatidate molecule. So what I'm going to draw is, I'll draw the molecule out in its own right. So let's draw another phosphatidate molecule here, and then it will have this phosphate group, and then I'll draw as a little blue triangle here just to represent that amino acid stuck on the end, which is the serine amino acid, which you have formed a phosphoester bond with the phosphate group of the phosphatidate molecule. Okay. And this is now what's meant by phosphatidylserine. It means serine linked to the phosphatidate molecule. So this is phosphatidylserine, PS. And I hope that what I've convinced you is that basically this is just a phospholipid with a extra group stuck on its head. So yes, it's going to have an extra bit sticking out into the cytoplasm or into the extracellular fluid, but it's still just a normal phospholipid. So phosphatidylserine is a normal component of the phospholipid bilayer. Okay, so basically, what happens is you will have these phosphatidylserine molecules in the phospholipid bilayer of the plasma membrane here. So let's draw one here. Okay, so here's our phosphatidate molecule, and then here is the modification to turn it into phosphatidylserine, and I'll colour it in again. So here is our hydrophobic tails in orange here. Here's our glycerol backbone in green. Here's our phosphate group in red. And finally our serine that's been linked by a phosphate ester link to that phosphate group, and we're doing that in blue. Okay, and what's going to happen is that when the calcium binds to that C2a domain, the C2a domain is going to bind to the phosphatidylserine here, which we'll abbreviate to PS. Okay, and that is going to, what it's going to lead to is it's going to lead to the whole of this plasma membrane sort of invaginating in to be near the C2a domain of the synaptotagmin, and that's going to pull the plasma membrane towards the membrane of the synaptic vesicle sitting here. Okay, so let's have our synaptic vesicle oddly shaped here, and that is what's going to lead to the fusion of these two membranes together. And I feel like I should draw a second layer for this synaptic vesicle. Since I've drawn the two layers of the phospholipid bilayer of the plasma membrane separate, it looks odd to not draw it separately for the synaptic vesicle as well. Okay, now this interaction between C2a and the phosphatidylserine seems to be the most important interaction between synaptotagmin and lipids. It seems to be the most important way in which synaptotagmin triggers the exocytosis of the synaptic vesicle contents. However, the C2b domain also binds to a lipid component of the phospholipid bilayer, and this may well contribute. It's not as important, we don't think, as the interaction between C2a and the phosphatidylserine, but we'll go through it anyway. Right, so which lipid does C2b interact with? Well, it's another modified phospholipid, and it's something with a longer name than phosphatidylserine. So again, you have phosphatidyl here, and this time it's our old friend phosphatidyl inositol 4,5-bisphosphate, which if you've ever studied any sort of cell signaling whatsoever, you will have heard of before, because this molecule is incredibly involved in the GQ pathway. Okay, so this molecule, because its name is such a mouthful, is often abbreviated to P for phosphatidyl, I for inositol, and P2 for bisphosphate. You've stuck two phosphate groups on it. Okay, so now let's try and understand what we have done, what this molecule actually is. Well, again, it's just a phosphatidate molecule, so again, it's just going to be a phosphatidate group, so let's draw one of these out. So here's our glycerol, here's our phosphate group, so let's colour everything in, the phosphate group in red. Okay, the glycerol backbone with its three hydroxyl groups in green, and the long chain carboxylic acids, which are esterified to the first and second hydroxyl groups of glycerol, in orange. Now, we have, that's our phosphatidyl group here, inositol. What is inositol? Inositol is a 6-membered carbon ring, like so. So it's a 6-membered carbon ring where all the bonds between the carbons in this 6-membered carbon ring are single bonds, okay? And where of every single carbon in that 6-membered carbon ring, you have a single hydroxyl group. So let me draw its skeletal formula down here. So here's the 6-membered carbon ring of inositol, and then off every single one of these 6 carbons, you're going to have a hydroxyl group, like so. Now, you will also have hydrogen atoms of every single one of these carbons to make up the 4 bonds that a carbon atom needs, but obviously you don't draw that in skeletal formulae, and it makes it look far more complicated than it needs to if you start drawing the hydrogens. So this is the structure of inositol. Now, it's got a beautifully rotationally symmetric molecule. Now here is the inositol. We're just going to colour it in blue here. We're not going to show all the hydroxyl groups off to keep our cartoon nice and simple. So here's phosphatidylinositol so far. So we've got this part of the name done. Now we need to add on 2 phosphate groups to make phosphatidylinositol 4,5-bisphosphate. Now, this carbon here is counted as the first carbon. Then you count round, 2nd, 3rd, 4th. Okay, so we'll add on a phosphate group to the 4th. 5th, so we'll add on a phosphate group to the 5th. Okay, so done. There's phosphatidylinositol 4,5-bisphosphate. However, if you stare at that molecule for long enough, you will think, wait a second, shouldn't we have called that phosphatidylinositol 3,4-bisphosphate? And I would agree with you. I do not know for the life of me why they called this phosphatidylinositol 4,5-bisphosphate rather than 3,4-bisphosphate. This is the structure of phosphatidylinositol 4,5-bisphosphate. But to me, it should be called phosphatidylinositol 3,4-bisphosphate. However, long ago, someone decided that the way that these carbons should be labelled is that you should label this carbon here with the phosphate group of it, the 5th one, rather than the 3rd one, which goes against all modern ways of naming chemicals, but never mind. This is phosphatidylinositol 4,5-bisphosphate. Right. Okay, so this is the lipid that is in the phospholipid bilayer, which the C2b component binds to when calcium has bound to it. So let's draw it in up here. So again, you'll have this present in the plasma membrane phospholipid bilayer. So here's our phosphatidate, and then we've got this 6... Oh dear, what's happened there? That's not good. I'll try and fudge it. There's our 6-membered ring. Okay. And here's our phosphate groups. It'll look fine once I've coloured it in. So let's colour in the inositol ring firstly to hide my mistake. Okay, there's our inositol ring. And here is our 3 phosphate groups in red now. Okay. Our glycerol molecule in green. And our long-chain long-chain carboxylic acids in green, orange rather, yeah. Okay, so what happens then? When calcium goes up in the vicinity of the synaptotagmin molecules, calcium is going to bind to the synaptotagmin 1 protein. 3 calciums are going to bind to the C2a domain, okay, and 2 calciums are going to bind to the C2b domain. Synaptotagmin is then going to bind to this snare protein syntaxin 1. Whether that's important or not, we don't know. It may well be important for getting the synaptotagmin close to the membrane so that it can now interact with the lipids, basically. Because now what's going to happen is C2b is going to bind to this phosphatidylinositol 4,5-bisphosphate, or PIP2, and C2a is going to bind to this phosphatidylserine. Together, what they are going to cause is this invagination of the plasma membrane towards, so it's going to invaginate like this. I don't want to draw it because it will ruin the picture. Now I can draw it down here, I suppose. It's going to cause an invagination like so of the plasma membrane, and that will bring these 2 membranes, the membrane of the synaptic vesicle and the plasma membrane, very close together, and that then is believed to be what causes exocytosis of the neurotransmitter that's within the synaptic vesicle on into the synaptic cleft. Okay, and of these 2 lipid interactions, the interaction between the C2a domain and the phosphatidylserine has a better claim than the interaction between the C2b and the PIP2, that it is the interaction which causes this invagination, i.e. this one's thought to be more important than this one. Okay, right, so that is what causes synchronous release of neurotransmitter, this rapid release of neurotransmitter when an action potential arrives. So when an action potential arrives, it's going to cause the release of all of these already docked vesicles from the presynaptic membrane here, and they're all going to release their neurotransmitter contents into the synaptic cleft, and that's this synchronous release of neurotransmitter. Okay, in the next video we'll talk about asynchronous neurotransmitter release.